They call themselves AIDS dissidents, and they count in their ranks a number of renowned scientists, including at least one Nobel laureate. They question the party line on AIDS, and they dispute most of what we've been told about its cause, about the safety and efficacy of AZT, and about the level of risk to the heterosexual population.
HIV cannot be the cause of AIDS because it doesn't infect enough cells and isn't active enough. It is a behavioral disease, it's not a contagious disease.
Recently, a growing number of voices have begun to call for a redirection and broadening of AIDS research. Is this the beginning of a scientific revolution within the AIDS establishment, or a noisy distraction from a massive campaign which, like the war on cancer, has spent billions of taxpayer dollars and has yet to provide a cure? This question is at the heart of the great AIDS debate. Hello, I'm Dara Wells. For the next 90 minutes, we'll debate and discuss key issues of AIDS research and policy with experts from all sides, but a word of extreme caution. Whatever you believe about AIDS, continue to practice abstinence and safer sex. Use a condom. What's at stake is your life and the lives of those you love. Now, let's introduce our panelists. Dr. Mark Kaplan is Chief of Infectious Diseases at North Shore University Hospital here on Long Island, New York, and a participant in the discovery of the HIV virus. Dr. Peter Duesberg is a professor of molecular biology at the University of California at Berkeley and a member of the National Academy of Sciences. Dr. Frederick Siegel of Long Island Jewish Medical Center has been in the trenches of AIDS research since 1979 and is credited as one of the co-discoverers of AIDS. Dr. Stephen Jonas is a professor of preventive medicine in the School of Medicine at the State University of New York at Stony Brook. Dr. Robert Gary is a professor of microbiology and immunology at the Tulane University School of Medicine in New Orleans. He is principal investigator of an HIV study now underway, funded by the National Institute of Allergy and Infectious Diseases. Dr. Richard Horton is North American editor of the British medical journal The Lancet. James Scutero is a freelance journalist. He has tested HIV antibody positive. And Joan Shenton is a documentary producer with Meditel, a television production company based in London. Joan Shenton, what does it mean to be an AIDS dissident?
It simply means that one is a critic of the orthodoxy, in other words, the view that HIV causes AIDS and that AZT is a useful drug for AIDS. What AIDS dissidents say is that HIV cannot possibly cause AIDS and that AZT is extremely damaging and toxic and of no benefit.
Professor Duesberg, what is your definition, or what is AIDS in your opinion?
I don't have a choice in that matter. AIDS was defined by the Centers of Disease Control for us, in fact for the world, as a symptom of now 30 previously known diseases that had all occurred prior to what we now recognize as an AIDS epidemic, but at a lower incidence. And those diseases have gone up in certain groups. That is what's perceived and defined as AIDS now.
Dr. Kaplan, what is the role of HIV in AIDS?
HIV is the cause of AIDS. HIV is a virus that's acquired either through sexual transmission or blood transfusion or perinatally from mothers. It's a virus which has a slow and indolent course of progression of destroying the immune system, particularly the T cell. And it ultimately leads to increasing destruction of immunity, which leads to a whole series of variety of infections that patients get who ultimately succumb to these infections. It's also a very potent neurotropic virus causing damage to the brain and also lets loose a lot of other agents within the brain as the brain's immune system is destroyed. And there's little doubt that it causes the AIDS epidemic.
We have a difference of opinion. To begin part one of our discussion, what causes AIDS, let's look at the orthodox point of view as summarized in this clip from AIDS Research, the story so far, produced by PBS station WGBH in Boston.
Scientists now know more about the human immunodeficiency virus than any other virus. It enters the body in blood or seminal and vaginal fluid and seeks out a key cell in the immune system. a white blood cell called the T helper lymphocyte, 150 times its size.
They have on their surface a specific doorway, a specific structure through which this virus can attach and enter. Like many unwanted guests, once it comes in your door, it decides to take its coat off and make itself at home, and that's what it does. So it enters the cell at the surface, it sheds its coat, and then it sheds its inner core, revealing its genetic material. In order for the virus to insinuate its genes within your genes, they have to be in a DNA form, not an RNA form. So the virus makes a DNA copy from its RNA base or template.
This DNA then could move into the nucleus of the cell and then become part of the cell's chromosome. So the virus, in essence, integrates itself into the cellular chromosome and becomes part of the cell.
The viral genes now take over the machinery of the infected T cell to reproduce itself. And then the virus migrates to the surface of the cell and buds out from the cell membrane. There's now a free new viral particle which can go infect an uninfected cell and start this whole life cycle over.
Before it is destroyed, one infected cell can produce thousands of HIV particles, which go on to infect other lymphocytes. Despite huge amounts of virus, all most infected patients feel is a bad case of the flu.
During the flu-like illness, there is enormous amount of virus in the bloodstream and probably in lymph node and other sites in the body. And in fact, the level of virus during this period is often higher than what you would find in a patient with terminal AIDS. But the immune system kicks in very quickly at that point and brings the virus under control in the course of just a few weeks. And it's very, very dramatic.
There's now a period where you feel fine. And that can last from a year or two, and then there are some people where it seems to last many, many years, a decade or more. Over this time period, though, the virus begins to chip away at the immune system. As the immune function falls below a level of 80% loss or more, people become predisposed to major infectious diseases. Pneumocystis pneumonia, tuberculosis-type organisms, toxoplasmosis, which causes an encephalitis, and so on. And they become predisposed to certain cancers, lymphoma and Kaposi sarcoma. Now, this was clinically called AIDS.
Professor Duisberg, that seems pretty cogent. What's wrong with the information we just saw there?
It didn't explain why HIV causes AIDS. It just pointed out that we have identified yet another retrovirus. We have done that in the last 30 years under the auspices of the virus cancer program with thousands of retroviruses.
All right, Dr. Siegel, I'd like you to answer that criticism that Professor Duisberg raised.
Well, for one thing, Dr. Duisberg says, and he's written, that AIDS is just a new name for an old disease. I've been working in immune deficiency diseases for 24 years. I've worked with some of the best people in the field. We know about susceptibility to disease, to secondary infections, to opportunistic diseases. And yet, when in 1979 and 1980, clinicians like myself who were trained in immune deficiency disease or infectious disease or in dermatology, who were familiar with what has been out there for the last or the preceding many years, began to see people with this new entity. It was so obviously a new entity that it did not take a genius to recognize that this was really a new disease. Let me finish.
Okay, but
I think we have to say something to it, because you give me credit for something or attribute me something that I haven't said. It is not my disease, and it isn't even a disease, it's a syndrome. S in AIDS stands for a syndrome, a collection of diseases, and I haven't said it's new. The Centers for Disease Control has said these are old diseases, and the appearance of them is new. That may be new, but none of the diseases is new. I haven't said that, and the CDC hasn't said that. If it's your impression, that's a subjective impression, but that's not what I said, not the CDC.
Have you not written that AIDS is just an old disease in new clothing?
That's what the CDC specifically stipulates. The definition says old diseases in the presence of HIV are called AIDS. That's not me.
Okay. But AIDS is a new immune deficiency disease, which was not present in this country or in Africa.
Well, it is not a disease. It's a symptom of 30 previously known diseases.
I'm sorry, but we have to disagree about that.
All right, Professor Duisberg, wait, okay,
Yeah.
let's go back to HIV itself. Do you believe that HIV is harmless?
It's totally harmless. Yeah, there are over 75,000 papers written on the subject with HIV. Not one of them proves that HIV causes AIDS. That gives you food for
Professor
thought.
Gary? Yeah.
But Peter, you know that HIV is not harmless. When it infects cells, CD4 cells, those cells die, or at least 90–95% of the cells
Dr.
die.
Gary, I have to respond to that HIV is mass-produced in T cells that have been infected by Robert Gallo and other researchers in the field. Now, 10 years ago, the very same cells are still growing in culture to produce that very virus that they're supposed to kill. They are immortal cells.
All right, let Professor Gary respond.
This is the phenomenon of persistence. This is what makes a retrovirus separate and distinct from other types of lytic viruses.
Totally agree with that, yes.
It has that capacity to undergo the reverse transcriptase reaction where it converts its RNA into DNA, integrates into the cell. But what sets HIV apart from several other retroviruses is that it kills many of the cells that it infects. It doesn't kill all of them, which is what...
I'm sorry, let me look at some of the photos that you brought, Professor Gary. Will this help us understand your point of view a little better? I think we're sort of talking back and forth here. All right, then let's take a look at the photos that you brought, and if you would describe them. I know it's a little difficult to see.
Well, this is just
a
colored version of the AIDS virus. It's a typical lentivirus, which is a type of retrovirus. We know in other animal species that lentiviruses cause neurological diseases and immune suppressive diseases, and that these viruses, these lentiviruses, can kill cells in culture. They're quite cytopathic viruses.
Okay, let's take the next slide then.
Okay, and so this is a picture I can see of the virus going into the cell at the plasma membrane. The yellow part is part of the plasma membrane of the cell, the outer barrier of the cell. And you can see even in this picture that just at that very initial impact of the virus on the cell that there's damage to the cell. There's perturbation to the membrane. You very often see the formation of pores, and the cell is damaged just by having contact with the virus.
So what Professor Duisberg says then, that HIV is harmless, is not the case as seen in those pictures?
That's right. That's exactly correct.
Well, I have to take issue with that. These pictures, of course, I can't deal with. They're not my pictures. You can always find a picture of a cell dying in culture. They always die in culture. In fact, T cells are very difficult to maintain in culture. But as Dr. Gary just pointed out very convincingly, the lifestyle of retroviruses is to become part of the cell. That was also said by Dr. Kaplan on television. That is the very essence of a retrovirus. That mechanism of replication only succeeds if the cell survives the infection. If it can enter the chromosome, if it can't, the virus is suicidal, it's dead. It cannot afford to kill the cell it wants to replicate.
So then
the cell has to survive.
So
that is the hallmark of retroviruses.
So you're saying then that a man who is HIV positive, he wants to have sex with a woman who is not HIV positive. Could you tell him that it's okay to have sex with her without using a condom if that's his only problem, that he's HIV positive?
You're saying that I'm saying that. I haven't talked about sex at all, but we can talk about sex anytime you want. I've talked about viruses. I've said that this virus does not kill cells.
Okay, but
and
you agree that
by its very
perhaps...
essence, it cannot kill cells.
Retroviruses
have been studied for 30 years prior to AIDS as the cause of cancer, all retroviruses, including HIV. It was called first the leukemia virus by Bob Gallo, who discovered it, among others.
Let me move to
because they don't kill cells. Only viruses that don't kill cells can also cause cancer.
All right, okay, this side of the table is getting a little restless, and then I want to go to Dr. Jonas. Yes, Dr. Siegel.
I think it's worth pointing out that in two situations, both in the human system and in experimental animals in the simian immunodeficiency virus syndrome, which is an analogous system, as I'm sure Dr. Duesberg will agree. Cloned viruses have caused disease in human beings and in monkeys. Ron DeRosier's experiment with a cloned SIV has clearly shown that these viruses are pathogenic and cause AIDS. The same is true of the accidentally exposed laboratory workers who got infected with cloned HIV. One of them already has AIDS, at least, perhaps you know how many others, but at least two and perhaps three are already symptomatic.
Well, Fred, I have two things to say to that. 150 chimpanzees have been inoculated with HIV. Not one of them has AIDS.
Isn't that very fascinating? And we'd like to know why.
So that contradicts what you just said.
No, it doesn't.
And the second point,
not at all, because this is a
the second point, I haven't said
virus.
anything yet. I had 2 points. You made 2 points. I answered the 2 points. The injection of the healthcare workers with cloned virus, whoever it was, has yet to be published. Fauci mentioned it on television.
This was the Nightline broadcast before
He also said
1994.
at the same time they were treated with ACT,
Yes.
which by itself is a cause of AIDS. It's designed to kill human cells, particularly fast-growing hematopoietic cells.
We can talk about AZT at a point later in the program. All right, we'll talk about AZT at a point later in the program. Dr. Jonas, can I get you in here? Is HIV harmless?
I don't know. I think that it probably is harmful. But my position, I'm a dissident as far as the conventional wisdom is concerned, but I differ. I'm a dissident with a different point of view from that of
What's
Dr.
your point of view?
Duesberg.
I'd like to hear it.
Point of view is that we don't know enough about either the epidemiology of AIDS and HIV infection, and that we don't know enough about the natural history of the development of AIDS to be sure either that HIV is the sole cause of AIDS or that it isn't associated with AIDS at all. My guess is, and this is a hypothesis based on really classic 19th century type purely observational epidemiology, my guess is that HIV is necessary but not sufficient to cause AIDS. That's my guess, but that's a hypothesis. Nobody is studying the hypothesis, as far as I know, from the epidemiological point of view.
I don't know if that's true. Didn't Dr. Lowe get research money for, what is it called, a mycoplasma, which is, Dr. Gary, I talked to your colleague, Dr. Voss, who tells me that this kind of research has been going on now for a while, that there is possibly a cofactor involved with HIV.
You have a problem again with the definition, which Dr. Duesberg often points out, that all diseases have cofactors. I mean, there are always complicating agents that can come into a person that has another disease and complicate that disease. With AIDS, with HIV disease, the fact that the virus debilitates the immune system opens the body up to many, many different kinds of pathogens, and some of these pathogens probably interact with HIV to accelerate the production of that virus and to get the disease process into sort of a cycle. So yes, it is true that some diseases and agents on top of HIV could accelerate the course of the disease, and in that sense they're a cofactor.
All right, Dr.
Well,
Jonas?
but I see it from the other way. See, I think that it's probably the other way around. And that is, I think that it's probably that if one gets an HIV, infected with HIV, and makes the antibody, and it should be clear to everybody, and it's not, that the test that we have is not for the virus, the test is only for the antibody. That the chemical, the chemical that the body makes in response to the appearance of the virus.
Could you respond to that, Dr. Kaplan?
I think that's a slight distortion of the fact. Originally, the test only measured the antibody. We have far more sophisticated tests nowadays that measure both the presence of the antibody. One can cultivate the virus from the bulk of patients who have this disease by using standard tissue culture mechanisms. One can demonstrate active RNA from the virus and even DNA from the virus by more sophisticated genetic mechanisms that we have available to us.
And what were those mechanisms? Is that PCR are you talking about?
In fact, PCR is one of the mechanisms of demonstrating viruses. There's actually southern blotting using a different methodology of detection.
There's P24 antigen testing?
No, P24 antigen measures the GAG protein of the virus, which is a protein of the virus.
There is a paper that's been published, and this was published in Biotechnology. And since I'm the only one at this table who has tested HIV antibody positive, I'm doing this for my life. I'm not doing this for a profession. So my life depends on this information. So first of all, this paper was published in Biotechnology magazine, June 11th, 1993, volume 11. It's a sister publication of Nature. It's produced by the same people. It was a front-page story on the Sunday Times of London, August 1st, 1993. What it's saying is that ELISA testing, Western blot testing, PCR, and P24 antigen testing is not synonymous with HIV infection, that the proteins that are detected by these tests are not specific and endemic to HIV.
Okay, could I get Dr. Horton to respond? You've been very quiet. You are the North American editor of The Lancet, a prestigious medical journal in its own right. What is your take on all of this? What causes AIDS?
There are two problems with the discussion so far. Firstly, the phrase AIDS. Dr. Duisberg is absolutely right. AIDS is something that we as physicians and scientists have concocted to suit our own purposes. Secondly, the word cause. We do know that HIV is associated with immunodeficiency, which is progressive over many years. The question that we don't know the answer to is, what does that association mean biologically? And that's what the focus of the discussion should be about. The discussion tends to get bogged down in, is this the cause or isn't it? That is not the scientific question.
All right, let's move on. Joan, I know you've done an awful lot of work in this area. One of the arguments you present in the AIDS catch is that many people with AIDS-like diseases, such as Kaposi sarcoma, are HIV negative. Is there anything we need to set this up, this next clip?
There are many thousands of people who have the full so-called AIDS syndrome who are HIV negative, and I saw many of them in Africa when I was there, and I know that they exist in the literature here in America, and that the CDC actually logs these cases of people with the full-blown AIDS syndrome, and they are HIV negative. Now this is very embarrassing to the AIDS orthodoxy, if I may say so. How do you answer this?
Well, we will let them answer it in just a moment. All right, yes. Let's now. Well, okay, they say it's not embarrassing, but let's now watch a clip from the AIDS catch produced by Joan Shenton.
Dr. Friedman Keene is convinced that his HIV-negative patients do not have AIDS, but a benign form of Kaposi's sarcoma caused by an organism other than HIV. Dr. Robert Root Bernstein has been making a special study of KS and believes that Kaposi's sarcoma of itself, without HIV, can produce an irreversible AIDS-type condition.
The existence of Kaposi's sarcoma patients who are HIV negative suggests to me that there are causes of AIDS other than HIV. In fact, I've just completed a study of Kaposi's sarcoma that goes back to the very first paper ever published on the subject by Maurice Kaposi in 1872. And it shows that, in fact, there are hundreds of Kaposi's sarcomic patients matching the CDC definition of AIDS for over a century. These patients are not elderly men. These are teenage boys. They are young men in their 20s and 30s. They are often described as being previously healthy.
The USA Center for Disease Control's definition of AIDS states that men under 60 with Kaposi's sarcoma who have not been tested for HIV or whose tests are inconclusive have AIDS. Would Dr. Friedman Keene's patients with Kaposi's sarcoma and no HIV be defined as having AIDS?
At this particular time, the CDC, the Centers for Disease Control, and other scientists and epidemiologists around the country are reconsidering the definition of AIDS to perhaps change the definition not to include Kaposi's sarcoma as a definitive diagnosis.
All right. Dr. Siegel, you said you wouldn't be embarrassed by this and you'd like to respond. How do you explain that?
Not at all. First of all, it's very clear that the surveillance definition of AIDS is being confused with definitions of AIDS that have to do with pathogenesis and immunodeficiency. Certainly Kaposi's sarcoma has been around for long before the epidemic of HIV, and whether you equate the epidemic of HIV spread with the epidemic of AIDS, as I do, or you argue that it is only a marker, it's quite clear that that epidemic, with the exception of certain interesting isolated cases dating back as early as the 1950s, is a new epidemic. Now, if you confuse the case definition as a surveillance definition, which both the Centers for Disease Control and the World Health Organization, with somewhat different sets of definitions because of feasibility, basically, with a definition of disease at the immunologist's point of view, then what you are doing is confusing apples and oranges.
I really
and you
don't
cannot
understand
really do
what
that.
you're saying, because fundamentally, if you can have full-blown AIDS without HIV, HIV cannot possibly be causing AIDS.
Well, that's
now,
a circular
you're talking
argument,
about
because if you
surveillance
say
definitions.
that Moritz Kaposi's first case of Kaposi's sarcoma had AIDS, then you are wrong. That fits the surveillance definition for AIDS, but it certainly
But why do you have to have a surveillance definition in medicine?
You need a surveillance definition.
What is a surveillance definition?
Before we knew what this disease was about, I'm an infectious disease person, Fred has been an immunologist. We suddenly saw a new type of presentation of a list of 30 infections that Dr. Duisberg made, tuberculosis, cryptococcosis, Kaposi's happened to appear on the scene to dermatologist's office. Cryptosporidium, a whole series of infections started to appear in presentations we had never seen before of these infections. So their presentation was different, and patients were not only getting one of these infections, but 5 to 7 of these infections. And some patients were getting serious neurological diseases that had never been seen before.
But some of these patients don't have HIV.
Some may not have had, because within...
So how can you be sure that this is the cause?
Let him finish,
Because
Professor.
there are lots of... Most infectious agents can slip into the immune systems of different people depending on the severity of immunity. But the multitude of infections that were seen in HIV patients, the presentation, we have never seen tuberculosis as it occurs in HIV patients. We have never seen cryptococcosis as it occurs in these patients. We have never seen cryptosporidiosis before the AIDS epidemic, really, in the presentations. So the definition fit nicely, and it allowed us then to pick patients who we said...
This is an artificial
it was an artificial construct.
This is an artificial construct,
Why
and
do
I'll
you
tell
say
you why.
that?
It was very artificial.
I'll tell you why, because
you.
the first 41 cases of AIDS in this country, according to the New York Times, where the article was Rare Cancer Found in 41 Gay Men.
Mm-hmm.
It was in the movie Long Time Companion.
I remember that headline, actually.
Right,
yeah.
you remember that?
Mm-hmm.
If you read the article, and the next 5 articles in the New York Times, every single one of the first original cases of people with AIDS were heavy drug users. They happened to be gay, but they were all admitted heavy drug users.
And I want to add to that, because you say you'd never seen this syndrome, but only this year, in fact a few months ago, one of the leading science journals, I can't remember which one right now, explained that drug addicts in New York who were HIV negative were showing the exact same symptoms as full-blown AIDS.
No, that's not correct either.
Well, how about Jackie Onassis? Jackie Onassis died from non-Hodgkin's lymphoma and pneumonia. If she were gay, she would have been given a presumptive diagnosis of AIDS. They wouldn't have given her an HIV.
Only if she were HIV positive, these gentlemen are saying. Okay, yes, Dr.
But she would have died of AIDS-like illness.
Dr.
How
Horton.
pointless it is to talk about AIDS. What we want to know about is what's the cause of the progressive immunodeficiency that you see that is associated with HIV infection.
Very good.
Let's forget AIDS.
Fine. Yes, Dr. Jonas.
And I think that the gap in the knowledge has to be filled in by doing the epidemiology and natural history study that hasn't been done. That's precisely right. In other words, both sides of the table could be correct. That what we have is we have something called AIDS. In some patients, it is associated with HIV, and in some patients, it isn't associated with HIV. What is the common factor? There has been a very serious figure ground problem in the study of this condition. The figure ground problem is, for example, when it was first identified in the gay male population in San Francisco, for a variety of reasons which we don't have time to go into in this segment, it was associated with frequent sexual relationships between gay men. That was the figure. I think we should look at the ground. It perhaps, in fact, I don't think it had anything to do with the frequency of the sexual relationships. I think it had to do with the diseases and conditions that gay men who engaged in frequent sexual relationships happen to have.
Such
chronic
as
sexually transmitted disease, hepatitis B, chronic bowel infection, and IV drug use. I think those are the factors which caused the weakening of their immune systems, so that they were, when they were exposed to the HIV virus, well, you can shake your head, Dr. Siegel, but the point is we don't know.
I'm sorry, but that doesn't square with the experience currently on Long Island, when both in my clinics and in Mark Kaplan's clinics, 45% of our patients are women, many of whom have had only a single sexual partner from
how many are drug addicts?
And it doesn't square with,
square
with our children. It's not anecdotal. We published a paper on AIDS in children with Dr. Savita Power. Now, I don't think our children were shooting cocaine or drugs.
Yeah, but what were the mothers doing when they were pregnant?
The mothers were doing when they're pregnant. But we've had lots of mothers who shot drugs, and we also had mothers who had blood transfusions.
There are also many smokers who don't have lung cancer.
Yes.
And there are more old drunks than there are old doctors. That's a specious argument.
There's 14 million people on this planet with HIV, all of whom are healthy, according to Mercer.
And they will all be sick, unfortunately.
No, that is a lie.
All right. At this point, ladies and gentlemen,
life is a fatal disease. I agree with you.
Yes, eventually all of
You
us
have
will
no
die.
epidemiological ground to say that someone who's HIV positive will develop AIDS. Period. There are
all I'm suggesting
excuse me?
Dr. Kaplan, all I'm suggesting is that we do the epidemiological studies that haven't been done, and I don't know, but they have not been done. I am sorry. There is no, don't throw that
in 1985, there were 1 million people who were estimated to be HIV positive in this country.
All right.
According to estimates now, 700,000 people are seen to be HIV positive in this country.
This is exactly where we are going, and that's why I want to interject at this point. At the beginning of the AIDS epidemic, one common perception, mistaken and discriminatory as it might have been, was that AIDS was a deadly disease that only affected people from groups that began with the letter H: homosexuals, Haitians, hemophiliacs, and heroin users. It wasn't until sometime later that we figured out that heterosexuals and really all human beings also met that criteria. Predictions about the scope of the epidemic have varied greatly, and some of the most catastrophic projections have simply not materialized, at least not yet. Now, our next area of discussion is entitled, Do the Numbers Add Up? And it begins as well with short excerpts from Joan Shenton's The AIDS Catch.
As the AIDS edifice begins to crumble, more anomalies emerge. UK government predictions, for example, said there would be 17,000 AIDS deaths by 1992. This has now been slashed to 5,000. That's by more than 2/3. And the number of predicted AIDS cases has been halved. Only a small proportion of HIV-positive people ever develop full-blown AIDS. In the USA, in any 1 year, only 1.5% of the estimated HIV positives get AIDS. This means that 98.5% don't get AIDS.
Joan, that is a short bite, but are there any more examples? Of the fact that so few people who have the HIV virus develop full-blown AIDS, according to the figures there in your program?
Well, I mean, most of the research for the 7 documentaries we've made about AIDS has actually come from Professor Duisberg, so I would like to ask you to answer that specific point.
Well, the discrepancy is growing. This is why the HIV establishment has invented something that's called the latent period. A virus that regenerates or replicates within 24 hours is said to take 10 years to cause a disease. That was invented because we had 1 million people acknowledged to be infected in the United States, but initially only 10 or 20 or now 50,000 of those developed AIDS annually, and 950 or 980,000 did not. So they were buying time by stretching the latent period. Initially they said, oh, in 10 months they get it. First they said in weeks, then came 10 months, that was the official number, in 1984, and now it is 10 years. They're buying time to allow something else to cause AIDS.
Doesn't hepatitis C, though, act the same way? I'm not a virologist, obviously, but...
Hepatitis C I cannot discuss. We don't know whether that's the virus. There's was no virus ever defined in hepatitis C. Hepatitis B is a viral disease. Hepatitis C is only a polymerase chain reaction. Nobody has told the virus.
Tuberculosis.
Well, let's stay with something that is...
But how about TB?
Yeah,
I mean,
just a
I
minute.
think...
That's terrific.
Just a minute.
Or herpes simplex
Let's
virus.
stay with the virus. If we go too far out, then...
If we go too far out, we're extending ourselves past biology into biology of infectious agents. And there are many infectious agents that know.
I was asked. Excuse me, just let him finish his thought and then
I thought I was asked to explain.
Okay. All I want to know is, are there other viruses that act the way HIV does, that they don't necessarily make somebody sick within the next month or even the next year?
There's not one virus that causes a disease only after it is neutralized by antibodies. And it's eliminated to a point that even leading AIDS researchers in the world, like Dr. Gallo and Robin Weiss, had notorious difficulties finding the virus. And there's not one virus that causes a disease or 30 diseases only 10 years after it's neutralized.
Really
not one anywhere in the world.
May I please hear from Dr. Horton?
I would love to see those examples.
There's a critical distinction here. Yes, the virus is eliminated from the peripheral circulation largely, but it is not eliminated from the body. The virus hides in 98% of lymphoid tissue, which is not the bloodstream. And very good work that's come out of the National Institute of Allergy and Infectious Diseases shows that it is in the lymph nodes where there is a progressive destruction of tissue, which leads ultimately to this immunodeficiency and then allowing superinfection with opportunistic infection.
The lymph nodes are the place where viruses are destroyed. All viruses and all antigens are brought to the lymph nodes.
And that's a critical question that we need to answer. You're absolutely right. We do not know the explanation.
You say the virus hides is giving it a life that it does not have,
but we don't.
A property. The virus cannot hide. It is not an inanimate or a piece of RNA wrapped in protein. To use terminology to say that it's a pathogen.
The virus lies in the lymph nodes.
No, it is brought to the lymph nodes by the lymphatic system.
Right.
But Dr. Horton, it is hiding there in 14 million people on this planet who are totally healthy, just as much. The key distinction is between a virus causing a disease and one not.
Answer your question. Okay, he says that they are healthy and they have the virus, and your response to that?
They are symptom-free, but their bodies are not healthy. They are having a progressive destruction of their lymphoid tissue.
According to what criteria?
According to the criteria of T cells?
Yes. T cells, I could show you 50 people with... I could show you 15 people with 0 T cells for 5 years who are perfectly healthy. CD4 counts are not an indicator of disease progression.
But we're not disagreeing here. No, we are not disagreeing here. What we're saying is that here is this virus in the lymph nodes. What we don't know is how it destroys the lymphoid tissue in the lymph nodes.
They don't even know how it fuses to a cell. You don't even know how it fuses to a cell.
We don't. You're right.
And you come up with excuses like apoptosis, which to me is the most ridiculous theory on... Yes, but this is the virus hides, the virus perturbs cells until they become hypersensitive, until they commit cell suicide. What kind of stories are these?
But the point
this is not science.
How do you know that it destroys these T cells in lymph node? How do you know that?
How does it do it by lysis, cell lysis? How does it kill the T cells?
It is a cytopathic virus, but we don't know how it destroys us. That's the
question. Excuse me, ladies and gentlemen, may I go to Dr. Right.
How do you know that it does? You say you don't know how, but how do you know that it does?
Could we go to somebody who's actually researching this? You mentioned the Institute of Infectious Diseases and Allergies. Dr. Gary.
They kill the cells.
Please,
how does it kill the cells? How does it kill the cells?
Can I talk to Dr. Gary, please? You're doing studies right now. Fine. May we please listen to somebody who's in the lab talking about this one at a time, please? We can't hear you all if you all speak at once. Let's hear Dr. Gary.
It is a very cytopathic virus in culture, and we can observe.
We went through that before.
And please let him finish, please.
Before. And this virus can perturb the cell membrane and cause damage to the cell membrane. The cells swell up, they lyse. It can cause 2 cells to fuse together. These cells, syncytia formation, those cells are not viable. And there are other mechanisms too. This virus probably has many, many different mechanisms that it's developed to kill T cells. Induction of apoptosis is one possible mechanism, but then it activates the immune system. Remember,
what do you mean by that? Please understand that our viewers are not physicians by and large or scientists.
Remember, the hallmark of this disease is the loss of the CD4 T lymphocyte. Okay, and this
Which is the
precisely
what?
the same cell.
Which is what? The T cell?
It's a helper T cell.
Okay.
It is involved in many, many important reactions in the immune system. It's said to orchestrate many things that go on in response to different invading organisms. And this is precisely the cell that HIV infects. Okay, so not only does the virus kill many of the cells that it infects, it doesn't kill them all, but it kills a good percentage of them. But the ones that survive the infection are set up for destruction by the immune system. The virus puts envelope proteins on the cell surface. That means that they'll be attacked by antibodies and by other cytopathic cells that are part of the immune system.
Okay, I flunked Chemistry 101, so I'm going to stop you at that point. But I want to ask Dr. Jonas, you know, I remember when AIDS, and it was AIDS that was announced in 1983 or 4, and I remember at the time that the Reagan administration was in office, they were somewhat, needless to say, homophobic. The disease first appeared in gay men. Is it possible that these figures and predictions of an epidemic were being put forth and manipulated somewhat because gay men were dying and they were afraid that if they didn't trump up the numbers a little bit or the predictions that people would just leave them to die and the government would leave them to die? Is that possible?
I think a false picture has been presented to the people of the United States about the AIDS epidemic, and I think it is an epidemic, whether it existed before 1978 or not. I think a false picture was presented, which was created politically by two sides of the argument, by both sides, by the homophobes in the Reagan administration who wanted everybody to look at AIDS as God's
punishment,
death blow punishment. Right. To those awful people, and the AIDS community, the certain elements of the AIDS community who wanted to say, yes, HIV is the sole cause of AIDS, because they wanted people to believe that it would eventually get into the heterosexual population, so that people would be scared enough so that there would be enough money given to AIDS research. I think that both sides made terrible mistakes and misled the public, and also misled large parts of the scientific community because of these political ideological
This is not an epidemic.
Can I come in on that point?
Yes, look at the numbers.
I would like to take that point up and follow it with a quote from Professor Charles Thomas, who's actually running the group for the scientific reappraisal of HIV. And there are 150 scientists involved in that.
Right, of which I and Peter Duesberg are both
yes,
members.
Indeed. He says AIDS statistics are manipulated to maintain the impression that everyone is at risk, which maximizes funding and fear, and to conceal the discrepancies between the HIV theory and the facts.
Let's look at the numbers.
Look at the numbers. Yeah, all right, fine. You get last word, and then I really do want to move on to something that I know you'd like to talk about in that. Okay, fine. Now, Dr.
Let's talk
Siegel.
about cohorts of people infected with HIV. Dr. Duesberg tells us that there are 14 million people in the world with HIV infection, and that only 1.5% of them has AIDS. I would make the rejoinder that if you look at the San Francisco cohort of men, or if you look at the cohorts of hemophiliacs who mostly acquired HIV in 1981 through 83, and you take as an example what happened to that group, we know that out of the 150 people at our hemophilia center at Long Island Jewish Medical Center, 50 are dead of AIDS. Half of that cohort is symptomatic, and the rest are asymptomatic.
Well,
that doesn't square at all with your statement, nor do any of the other cohort studies that have been done and published within the United States.
No, let me respond to this.
Please do, and then we will move on to our next subject.
This fits exactly with my theory that AIDS is not caused by HIV, but another factor. And that is associated with those risk groups that you just identified, namely male homosexuals, hemophiliacs who are recipients of foreign protein transfusions for a long time in order to sustain their lives, and intravenous drug users. Those have been and are the risk groups in AIDS ever since AIDS existed in the United States and in Europe. And that is the group And that is the group that gets AIDS. The others who are infected by HIV do not get AIDS. That's 97 to 98% of our AIDS patients.
This is a very dangerous concept,
just
which I think must be refuted now.
Excuse me. Why is it?
I was trying to explain my others.
It's dangerous to you.
All right, yeah, finish your sentence, Professor.
For people to believe that just because they're not in a risk group, that they are not at risk.
Excuse me, let Professor Duisberg finish his thought, and then I would like to have a response from Dr. Kaplan, and then we really do need to move on.
Can we just look at the numbers?
We only have 90 minutes. All right, Professor,
97% of the American AIDS patients, according to the CDC, are either intravenous drug users, male homosexuals, or recipients of transfusions. I deduce from this that this is not a that's not an infectious disease at all, because infectious diseases spread in the general population, exactly as Gallo, the Surgeon General, Margaret Heckler, the Department of Health has predicted it, and that hasn't happened. And I'm looking for another cause, and these causes I will identify are the drug use in the male homosexual and the intravenous drug use communities, and the transfusions, the foreign proteins that are
I see you're going over ground you've covered before. I said, Dr. Kaplan.
There's a point that hasn't been covered before.
We're not allowed to give our point, and I think we should.
Please do, Dr.
First of all, amongst the hemophiliacs who've gotten the disease and those who have not, there are a group of hemophiliacs who've tested HIV negative, and they have not developed AIDS, in spite of the fact that they're exposed to the same amount of proteins. Second, there are many HIV negative drug users who continue to shoot drugs. They do not get the degree of infections. They may die of overdoses. They may die of hepatitis B. They may die of murder and so forth. But they're not. If they're HIV negative, they're not getting the same cohort. We have many. We have many women. We have many women out there now.
Look at any crack addict. Look at any crack addict. And you're telling me they're not dying.
Okay, we're losing control here. All right.
Absolutely. You are unaware of the literature. There are 18 studies that I can document. You have me and my
we will move on now. You're going to spend the entire rest of it.
The editor from Lancet has published the bulk of the best epidemiology.
All right, last word, and I do mean last word on this from you, Dr. Horton, and we do need to move on. We have a lot of ground to cover. Now, finally, if drug use is the cause of AIDS, why are we seeing AIDS now and not back in the 60s when an awful lot of people were using drugs?
We saw it then. That's exactly right. No, that's because we have a progressive decline in the immune function. That is the critical point here in this discussion, which we're forgetting in all this discussion about what is AIDS, what's the role of HIV. Somebody becomes infected with human immunodeficiency virus.
HIV.
Right. There is then a progressive decline in their immune function.
Is not true.
That is true.
Blatantly not true.
It is. It's not. It is.
Now, what I'm saying.
No, a million people.
Let me finish. Let me finish my point. My point is this. I am not saying that there is cause and effect there. There is not cause and effect that's been proven. But HIV is necessary in order to get that progressive decline in immune function.
It's not true.
My belief, and many others believe this, there are other factors that determine the progression. That is why you have long-term survivors who are HIV infected.
All right, very good. We're going to turn our attention now to treatment issues and specifically AZT. The following excerpt comes from Joan Shenton's Diary of an AIDS Dissident.
So AZT was developed in the 60s. John Lawrence already gave you part of the story, to kill human cells. That was what it was developed for. It was a designer drug to be used in cancer chemotherapy. It is an analog of the 4 building blocks of DNA. Thymidine exactly is the building block that it is related to, and it acts as a chain terminator of DNA synthesis. So whenever AZT is incorporated into DNA instead of thymidine, the DNA stops right there. That's the end of the DNA. And when DNA stops, the primary consequence is the cell is gone. DNA is the central molecule of life, and if you terminate it, then it's time to say goodbye to this planet.
Duisberg went on to criticize earlier studies by Gallo, Broder, Furman and others that led to claims that AZT can target HIV and is 1,000 times more toxic to HIV than to healthy human cells.
If the drug is more toxic to the agent that is presumably causing disease than it is to the host, that is what they call the pharmaceutic window, or the therapeutic window in which you can operate.
Toxicity levels and dosage recommendations in the doctor's reference books are still set according to the earlier low toxicity findings.
But in the meantime, a number of other investigators have also checked the toxicity of AZT on cells and viruses. And they found that Dr. Gallo erred and the Boris Wellcome erred by a factor of 1,000. That is not a minor little error. They found that the toxicity for the cell was a micromolar. It's still based on...
This makes it 1,000 times more toxic than was originally claimed.
So Gallo, as I said, faithfully believed, and so did Boris Wellcome and Broder, that AZT therapy was justified because AZT is 1,000 times less toxic to the cell than it was to the virus. So that belief clearly must be questioned now, because experimentally that result could not be reproduced by 4 independent studies.
Duisberg then moved on to attack the studies that led to the licensing of AZT. He pointed out that figures for deaths showed 19 in the non-drug or placebo group and only 1 in the AZT group. But 30 of the AZT group suffered such severe anemias they had to be kept alive with multiple blood transfusions. This was the case in only 5 in the placebo group.
They said in the same paper, if you look at the table, that 30 guys needed multiple transfusions. That is a euphemism. They would have died from anemia, that is, they didn't have enough red cells to breathe any longer, had they not been transfused with red cells from somebody else to keep them alive. So if you figure that in, and in the placebo group there were only 5, and it is quite possible that those in the placebo group get the anemia from sharing AZT, because they figured out very fast who got AZT and who got placebo. They taste different and they smell different, and they actually did patient-initiated drug sharing. That could be the explanation for the anemia in the control group as well. So if you add that up, if you include the would-be deaths, had it not been for the transfusions, we would have 31 would-be deaths in the RCT group and 24 in the placebo group. And now there's no longer any reason for celebration.
Dr. Kaplan, I watched you shaking your head during that. Why?
I wasn't even listening to the AZT argument because I have my own misgivings about AZT, and whether Dr. Duisberg believes it or not is irrelevant to me, or Ms. Shenton believes it or not. I'm trying to figure out how to counter the arguments that if we see somebody hit by a truck and we know it's been driven by a drunk driver, and I try to stop the drunk driver and he still kills the person, it doesn't mean that the person wasn't killed by the truck if I couldn't stop the truck. And you know, if I knock over this glass and the water spills out, I can give you a million reasons why my hand moved over, it banged the glass and it spilled over. Just because I can't give you all the intricate reasons of why that happened doesn't mean that it didn't happen. When I see my AIDS patients suffering and dying and I measure their immune function and I find it's been destroyed, and I've known some of these patients over 10 years and I've seen a steady, slow progression of their disease, I've tried to intervene on their behalf to help them with the drug AZT, which I was hoping might be very beneficial, and I still think in certain circumstances has been, doesn't mean that we're all wrong about it. There are certain utilities to that drug. It's only a drug, and we all know it's got its difficulties, because
we are still wrong about AZT.
So you're wrong and I'm wrong, and we're not stopping.
Okay, there are famous studies that are now made. The Concord study has just shown that there is no benefit from AZT in asymptomatics. And there's another famous study by your own people at university here in North Carolina, which say that the deaths of the people who were less sick were accelerated by
All right.
AZT.
Can we save some time
why
here?
do you think AZT is useful? How can you possibly
I didn't
that?
say it's useful.
Can we save some time here? Is there anybody here on this esteemed panel who feels that AZT is the drug to use to try and save AIDS patients? And if it's not AZT, what is it?
I think that this has to be addressed because it's always raised.
Right.
There are very large statistical studies, including the Concord study that's just been cited, that compare placebo recipients to recipients of AZT. None of those studies shows the averred toxicity that Dr. Duisberg and his colleagues are insisting on. Indeed, hear me out. Hear me out.
The data is
The
there.
data on hematological toxicity and blood transfusion is in the paper.
No, I'm sorry.
He's
but
reading the Lancet.
Yes,
the paper's here.
Whose study was that, Dr. Horton?
This is the Concord study that was published in April of this year, an Anglo-French study, the largest, the longest study to look at the efficacy of AZT in symptom-free HIV-infected individuals. And there was substantial hematological toxicity, which led to patients having to withdraw from the trial, and not only that, requiring blood transfusions, which, as Dr. Duisberg pointed out...
But you didn't let me finish my point.
All right, Dr.
Which is that in order to analyze efficacy or toxicity of drugs, we use Kaplan-Meier analysis. Kaplan-Meier analysis, there is no doubt that this drug, like every other drug we use in medicine, has toxicities. And a thinking physician doesn't follow a drug protocol the way the protocols are written. We adjust the dose. We do what we stop the drug when the patient is intolerant. We do not cause disease if we can avoid it. When you do a clinical trial and you have to follow standard procedures and not make dose adjustments and follow a protocol, it's more evident that these toxicities exist. But certainly neither Mark Kaplan nor I would quarrel with the idea that this drug has its toxicities, but so does aspirin.
Okay.
Nevertheless, if you look at Kaplan-Meier survival, the drug survival has never been worse. Never been worse than the placebo. So that whatever the toxicities are, and they're there,
Excuse me, may I talk to somebody who has tested positive for HIV antibodies? James, you are not taking AZT?
I never took AZT.
And why not?
And can I tell my story here? And we can talk about the experts who have been murdering their patients. December.
Well, that's a little extreme. Why don't you just talk about from your point of view? Because yes, I would like you to absolutely so, but I'd like you to express it from where you're coming from and what your situation is.
From exactly the same point of what I have seen, because I have a PhD with living with this diagnosis. December 18th, 1989, I was diagnosed HIV positive. I had just finished a 109-mile bicycle race in High Point, New Jersey. I was in the prime condition. I was president of my own computer consulting corporation, and this floored me. They told me I was surrounded by 3 clinicians in their white robes, in latex, telling me if I did not take AZT, because my T cells were at 336, I would die. I told them to go to hell. And 4.5 years later, without any medication, and despite my CD4 count being 219, I still rollerblade around Central Park, 18 miles. I am in fit condition, and all I have seen is lies and nonsense from the medical community, because Burroughs Wellcome, the maker of AZT, underwrites all the AIDS conferences. The Berlin AIDS conference was underwritten by Burroughs Wellcome, Abbott, and Bristol-Myers Squibb. The next AIDS conference is in Tokyo. Who the hell can afford to go to Tokyo?
So, James, you're saying that
the pharmaceutical companies are leading these doctors, these non-thinking doctors.
There's a conspiracy at work here?
No, there's no conspiracy here. This is an opportunity to make money. This is
Dr.
an opportunity.
Gary, would you respond to
Why
that,
have your
please?
T-cell counts dropped from 300 to 200?
I'll tell you exactly why.
300 is a very low number for CD4 cells.
Yeah, then how come I have not experienced any disease in the last 4.5 years?
Your remaining cells are working overtime.
This is nonsense. This is fantasy. I know people.
It's not normal for your CD4
Don't
counts
give me
to
this
drop from
normal. I
300
have
to
just done a
200.
study on T cells. I have pulled 17 studies of low T cell counts in healthy individuals. Russian powerlifters with T cell counts of 237 CD4 counts. There's a group in New York called HEAL. Health Education AIDS Liaison, where people get away from these murderous swine, get off their medication, and are thriving without
All right,
AZT
James.
or DDI or DDC.
I understand that this is a very emotional
No,
issue
this is
for
coming
you.
from knowledge, ma'am. It is not coming from emotion.
But I don't think
see
you have
every
to be.
week
I don't
people
think you
getting
have
off
to
their
be quite as
medications
rude as you're being.
and thriving. And my own
It
personal...
is very frightening to see your friends given AZT actually begin to die in front of you, because AZT is chemotherapy, and it is
No,
attacks
but
the
no
bone
one,
marrow.
but no
I
one
have
answered
the
my
side
question here.
effects of
Please, could
AZT
we
right
just
here.
talk about, is there another way to go? Even if AZT is not the answer, and look, let's face it, people were dying, people were scared, they were begging for something. Maybe AZT was not the way. Is there, Dr. Gary, is there a way to go?
Follow. I mean, the most important thing from this part of the discussion is it doesn't follow, it's not logical that even if AZT is not a perfect drug, and everybody admits that it's not a perfect drug, that's why we're doing so much work to try to find better ways to treat AIDS, HIV disease. But it doesn't follow from that just because AZT is not a perfect drug, that HIV is not causing the immune deficiency that we cause AIDS.
You asked, is there another path that we could
Please, and I
follow?
still haven't heard it.
The answer is yes. First of all, the more we've pursued HIV, unfortunately, the less successful we have been. When you use even more specific drugs that target the RNA and DNA involved with HIV, you do not have substantial efficacy. But if you try and stimulate the immune system, and this is very exciting work that's being done at the National Cancer Institute by Gene Shearer, then you may well have a possibility of boosting immune function and overcoming the progressive decline.
And this leads us to our very next topic, which is research. While both sides have theories as to what causes AIDS, nobody has come up with a solution for stopping it, despite 13 or more years of trying and billions upon billions of dollars of public money spent on the problem. Now, lawmakers in Washington are talking about a possible Manhattan Project for AIDS, yet it would appear that we're still fighting the virus with sticks and stones, and that leads us to ask, are we on the right track? Dr. Gary, are we on the right track as a researcher?
Well, if the right track is to attack HIV, which is the cause of this immune deficiency, we're on the right track. Now, the questions that have been brought up recently and actually by many of us behind the scenes over many years is, are we spending enough on basic research?
Right. I believe Dr. Fields of Harvard raised this very issue.
Bernard Fields, who is one of our most prominent virologists. He's written a definitive text on virology, and he recently published an editorial in the journal Nature saying that we do need to go back to some of the basics of virology and look. And there are many things we don't know about this virus. There are many things we don't know about the disease, and that if we take a step backwards and begin to learn more about it, which I think everybody could agree around the table here, then we'll have better ways to combat it.
Professor Duesberg, I understand you were going to have a meeting with Dr. William Paul, who heads the newly formed Office of AIDS Research that the federal government has established. Did, in fact, that meeting take place?
That took place yesterday.
And what happened?
Well, in that meeting, I was seeking his advice to find funding to test alternative hypothesis on AIDS, because the current virus hypothesis has cost the American taxpayer alone $22 billion in 10 years. It has not saved one single life. It has not produced zero public health benefit. It has been a total failure in terms of public health benefit. It hasn't made one correct prediction.
What kind of test or experiment would you do if you got the funding?
I was going to test a very testable and cheap hypothesis, namely that what affects the majority of our AIDS patients, recreational drugs and ACT. Recreational drugs, like intravenous drugs, that accounts for 1/3 of all American AIDS patients, or over 100,000. Most heterosexuals with AIDS are intravenous drug users. 60% are male homosexuals who have been, just at a meeting that also occurred at Gaithersburg yesterday, documented to have used recreational drugs like nitrite inhalants. I was going to test the effects of these drugs on animals.
And are you getting the funding, or do you know yet?
That is difficult. He said he would see what he could do to help me, but he hasn't promised it because he represents the HIV establishment, which is the majority of American
Well,
science.
I think you're just deducing that. You haven't heard his answer yet, and I think you should wait and see what he thinks. Dr. Horton, what's wrong with that, though? What's wrong with testing something like drug use as a possible risk?
There's absolutely nothing wrong, and in his article, Bernard Fields identifies the fact that we have been pursuing increasingly the wrong track, and that track has been to focus purely on the virus to the exclusion of other hypotheses. That's what we need to break away from. We do need to broaden the basis of AIDS research, but the problem is this huge infrastructure, this industry that's associated with the virus, and that's what we have to break apart.
It's hard to move direction, isn't it?
It's very hard. Let me give you one
quickly,
example.
And then I want to move on to our next
hard.
segment.
We're about to spend $50 million on phase 3 trials of vaccines to prevent HIV infection. Phase 1 and phase 2 trials give very little indication of efficacy. It's a waste of $50 million.
And it's a shame.
But can we reverse it?
That's the question. Now, one area of research that's gaining interest for obvious reasons is the study of long-term survivors. Now, this excerpt is from the PBS WGBH documentary, AIDS Research, The Story So Far.
In 1987, an administrator at the Sydney Red Cross Blood Bank, Jenny Learmont, got a call about a 75-year-old man who had discovered he was HIV positive. He had received a blood transfusion 4 years earlier. Today, all the blood taken in by the Red Cross is tested for HIV. But in 1983, when the man received his transfusion, no such test existed. So Jenny Learmont checked through the records to find out whose blood might have infected him, and discovered that he had received 1 unit of blood from a young man who was later diagnosed HIV positive. Over the next few years, several other recipients of this donor's blood turned up. All were HIV positive. All expected to develop AIDS and die. But as time went by, she realized an extraordinary thing about this group of 6 patients. None of them seemed to be developing clinical AIDS. In fact, their health was fine. Dennis, a 65-year-old man, was transfused in 1984 during gastrointestinal surgery. Like the other members of the group, he didn't want his face shown because he fears the stigma of friends knowing he is HIV positive. Despite the fact that he has been infected with HIV for 10 years, his immune system is in perfect working order. The same is true for the other recipients of the infected blood, like Marion, a 40-year-old woman transfused in 1984. Jenny Learmont realized she had stumbled on a group of people of immense importance to AIDS research.
There's 3 men and 3 women. They're very different ages. They range now from nearly 80 down to 40. They had their operations were all different, so they were transfused for different reasons. So the only common factor about that group is that they received blood from one person. The group themselves are all really well. Their whole immune picture is normal. And they've been infected now the longest 13 years and the shortest 10.
Jenny and her colleagues believe that the donor, who is also in perfect health, must have contracted a weakened strain of HIV, and this is being checked in the lab. If this turns out to be true, the group has unwittingly participated in the first live attenuated HIV vaccine experiment.
Okay, Dr. Kaplan, would you respond to that, please?
Yes, this study on long-term survivors is an extremely important study. In terms of research dollars, the peculiar thing is I agree with Dr. Duesberg on this thing. I would give Dr. Duesberg the money to study what he wants to study. Why? He has so much energy to come to these tables to get the money. I'd give it to him because Dr. Duesberg has made major contributions to our understanding of retrovirology. Let him have it, and in his way, he'll do something. I would ask him at the end of it, of the study, to do as he had done up to the early 80s, to publish that data for us in a scientific journal, refereed, call the referees if you want, and then let us see what he comes up with and see if it fits with the hypotheses. In other words, we don't need to be restricted. We don't have to assume the result will be as we'd like it. Sometimes in a disease which is not, I still think HIV causes the disease. There's no question about that in my mind. But I still think Dr. Duesberg and others of his type should be allowed to study this virus, as crazy as it may seem.
So if you were asking for the money from Dr. Kaplan, you would get your grant.
Yeah,
I'd give it to you.
I had just written down I was going to invoke the scientific method. And that calls for alternatives when a leading hypothesis is unproductive. And Mark Kaplan has done it for me. I couldn't agree more with him.
All right, very good.
I mean, that discussion is productive.
Yes, the other side of the coin is...
The other side of the coin is what?
The other side of the coin is, though, you have to also allow funding for those people who believe that HIV is the cause of the virus, and they will... Dr. Horton
will
publish...
I'm not worried about them yet. They get $6 billion a year.
He just wants a little, Dr. Kaplan. He wants a little.
I have some dissident ideas maybe on how the virus works. How do I appeal to Dr. Horton's journal? We'll send it into Dr. Horton's journal, and he then has referees who he chooses, and let the referee... That's science. And the fact about HIV, the fact
about...
a
long time. Can I just...
No, I want to just point out that everybody here is making deals on this program. When did this start happening? Dr. Horton, publish my article, and I'll give you some money. Yes, Joan.
Over 7 years has not been allowed to publish in scientific journals. Do you know that Nature has never allowed one piece from him in it, and they have consistently attacked him? Did you know that the New Scientist has persistently
I can't get into Nature. I wish I could.
Did you Dr. Horton, excuse me,
there is no voice for the AIDS dissidents.
We're getting a little bit into the media treatment, Joan. You're jumping the gun a bit, but I want to ask you, is Professor Duesberg being censored, or is it simply that he doesn't have the data to back up his theory?
I think both of those answers are correct. I'm afraid to say that some of my editorial colleagues on other journals have operated censorship very openly and have admitted that they will not publish his point of view. Indeed, they will go as far as they can to publish points of view that directly contradict Dr. Duesberg.
And ridicule him.
All right. Now,
Can I make one point on long-term
survivors?
research?
Yes, and
on long-term survivors. A while back, Dr. Siegel uttered 3 very important words: San Francisco cohort. That's exactly my point. Much of the conclusions that have been drawn over the years refer to the San Francisco cohort of gay men. The San Francisco cohort was drawn from two previous study groups. One of them came from a hepatitis B clinic, and the other one came from a chronic sexually transmitted disease clinic. That is exactly my point. We must look at long-term survival of people who have been infected with the HIV virus and have HIV antibody and have nothing else.
Okay, now with the focus of research beginning to shift, let us shift our attention now to the topic of the media's and scientific community's treatment of alternative points of view. We did talk about it briefly, but once again, here is a clip from Joan Shenton's documentary, Diary of an AIDS Dissident.
One of the first questions asked at the opening press briefing was why had Peter Duesberg not been invited? The conference chairman, Professor Otto Habermehl, said it was because he hadn't presented a paper to the selection panel, which is nonsense, because neither did any of the other key speakers before they were invited. Interesting to note that Habermehl also said that not everyone who has HIV will progress to AIDS, a major shift from the early days when a death sentence was pronounced on everybody with HIV. At the opening press briefing, Professor Habermehl had described people who question the role of HIV as mentally disturbed groups. Celia Faber took up the cudgels, reminding the panel that many distinguished scientists are questioning HIV.
Would you refer to Dr. Mullis, who's one of your colleagues, as mentally unstable, which is what you actually did? If you were not aware of this, now that you have been made aware of it, would you consider an apology?
I think it was really not a good way to say mentally disturbed, but I would change it. I would say bizarre.
I was denied the opportunity to ask a question, so tried to interview Professor Habermehl at the end. You said that you had sent this person out of the conference because he hadn't paid for the booth. Why did you allow 5 other organizations who had...
He just walked away.